Hello guys, welcome to my YouTube channel. This is me, Aniket Gupta. So today I am going to discuss with you a very important topic, that is bacterial genetics. And in this topic we will talk about the genetic recombination in bacteria, right? The recombination systems, the genetic recombination systems in bacteria, as we know that. And they are like conjugation, transformation, transduction, right? So these all three topics we will discuss one by one through these video lectures. And also, you have not subscribed my channel yet, please subscribe and press the bell icon, and also share the link of this video with your friends, right? So let's start. As we know that bacteria are the prokaryotic organisms, they have been considered as a simple organisms because they lack the cellular complexity as that of eukaryotes, right? Eukaryotic cell possess a well developed cellular organelles, right? But if we talk about bacteria, they lack all the cellular organelles, right? And bacteria are extremely diverse, and they come in variety of shapes, variety of sizes, right? Some are rod shaped, whereas some are spherical, right? And most of the bacterial genomes consist of a circular chromosome, and that circular chromosome contains a single DNA molecule having several million base pairs in length, right? So there is a single DNA molecule in the bacterial genome. For example, if we talk about the E. coli, that is Escherichia coli genome, it has approximately 4.6 million base pair of DNA, right? And some bacteria contain multiple chromosomes as well. For example, Vibrio cholerae. Vibrio cholerae is a bacteria that causes cholera, that is a bacterial disease. And this Vibrio cholerae has two circular chromosomes. If we talk about the Rhizobium meliloti is a nitrogen fixing bacteria which is found in the root nodules of leguminous plants. And this bacteria very beautifully fix the atmospheric nitrogen, and it has three chromosomes, right? And there are even a few bacteria that have linear chromosomes as well, and many bacterial chromosomes are organized very efficiently, right? More than 90% of the DNA in the E. coli encodes proteins, and in contrast about 1% of human DNA encodes proteins. So you can see this large difference, right? So this is the chromosomal DNA of a bacteria. You can see the very expanded form of this chromosomal DNA, and you can see also here this is a plasmid, and these all thread-like structure referred to as chromosomal DNA, right? Now we will talk about the plasmids, right? Many bacteria possess plasmids. Now what are plasmids? These are the small circular DNA molecules which is present in bacteria, right? And some plasmids are present in many copies per cell, whereas others are present in only one or two copies. In general, if we talk about in general, plasmids carry genes that are not essential to bacterial function, but they may play an important role in the life cycle and growth of their bacterial hosts, right? The great importance of plasmids are they are used extensively in the genetic engineering processes, and some of them play a role in the spread of antibiotic resistance among bacteria, right? Because they contain some genes which are resistance to which show resistance to some antibiotics, right? So these are the importance of plasmids. And plasmids जो है, these are the small circular DNA molecules. So you can see here the electron micrograph of plasmid. This is a plasmid, and the strands separate at ori. Ori, this is the origin of replication. The strand is going to separate, right? Now, most of the plasmids are circular and several thousand base pairs in length. Although plasmids consisting of several 100,000 base pairs have also been found, right? And each plasmid, if we talk about the structural arrangement of plasmid, so this plasmid possesses an origin of replication, right? Origin of replication, a specific DNA sequence where the DNA replication is initiated. So what is origin of replication? A specific DNA sequence where the DNA replication is initiated, and this origin allows a plasmid to replicate independently of the bacterial chromosome. So bacterial chromosome, independent of bacterial chromosome, this plasmid show replication, right? Now another term is there, episomes. What are episomes? Episomes are the plasmids that are capable of freely replicating and are able to integrate into the bacterial chromosomes, right? The F factor of E. coli, that is fertility factor of E. coli, is an episome, and that controls the mating and gene exchange between E. coli cells, right? So this episome simply what they do, they promote the gene exchange between the bacterial cells. So this is the F factor. You can see over here some of the genes are shown over here. These genes regulate plasmid transfer to other cells, and that is D, S, G, H, F, N, U, C, B, K, E. These are the genes, right? Then ORIT, that is origin of transfer. Then Rep, Inc, these are the genes which control plasmid replication. Then ORI, that is origin of replication, from where actually the replication is initiated. And then IS3 and IS2, these are the insertion sequences which regulate insertion into the bacterial chromosome. So this is the F factor, a circular episome of E. coli, a number of genes that regulate transfer into the bacterial cell, replication and insertion into the bacterial chromosomes. Now we are going to discuss the genetic exchange, that is bacterial exchange genes through conjugation, transformation and transduction. The first we will talk about the conjugation that takes place when the genetic material passes directly from the bacteria to another. There is a close association between the two bacterial cells. The one is the donor and another is the recipient. The one which donates the plasmid or a part of chromosome is refers to a donor, and the other which receives these things are referred to as recipient, right? So subsequent to conjugation, there is a crossing over takes place between the homologous sequences in the transferred DNA and the chromosome of the recipient cell. In conjugation, DNA is transferred from only donor to recipient with no reciprocal exchange of genetic exchange, right? And you can see over here, these are the two cells, two bacterial cells. One is donor cell and the other is recipient cell. There is a cytoplasmic bridge form, and DNA replicates and transfer from one cell to other, that is a red DNA, and this transfer from one cell to another, and transfer DNA subsequently replicates and crossover in the recipient cells leads to the there is a crossing over takes place, and then creation of recombinant chromosome, right? So this process is conjugation in short. and in other video lectures we will discuss this topic in very much detail. Next is as transformation. Transformation takes place when a bacterium takes up DNA from medium in which it is growing, right? So in which a medium in which the bacterium is growing, that bacterium will take the DNA from that medium. So after transformation, recombination may take place between the introduced genes and those of the bacterial chromosome. So you can see over here DNA fragments get inserted into the bacterial cell, then crossover takes place, and then there is a creation of recombinant chromosome. Next is a transduction that takes place when bacterial viruses, that is bacteriophages, those viruses that infect the bacteria, carry DNA from one bacterium to another. Inside the bacterium, the newly introduced DNA may undergo recombination and the bacterial with the bacterial chromosome. So you can see over here as shown in the diagram, there is a virus attaches to the bacterial cell on the cell wall, then injects its DNA, then once injects its DNA, the bacterial chromosome gets fragmented into fragments, broken into fragments, and that fragment will go and combine with the DNA which comes from the virus, and then virus will take that recombinant DNA, and then there is a crossover formation in the recipient cell, and then finally there is a creation of recombinant chromosome. So this is all about for this video lecture, and in next video lecture we will discuss these three processes in detail. So before that, let's check your concept, right? So please answer this question in the comment section, right? So which process of DNA transfer in bacteria requires a virus, right? So कौन सा ऐसा प्रोसेस है जहाँ पर डीएनए की ट्रांसफ़र करवाने के लिए बैक्टीरिया में वायरस की रिक्वायरमेंट पड़ती है, नीड पड़ती है, रिक्वायरमेंट होती है वायरस की. So if you know the answer of this question, please comment, please give the answer in the comment section. So thank you guys, thank you very much.